So today in class we're going to talk about the third type of passive cellular transport. If you recall earlier, last class, we started discussing facilitated diffusion and just regular diffusion as the first two forms of passive transport. But I said we would wait and talk about osmosis next class. All right, so osmosis is our third type of passive transport, which means, again, that there's no energy involved in moving the solvent, in this case we're talking about water specifically, across the membrane. Okay, so since it's passive, no energy is being used by the cell. All right, now diffusion is specifically, osmosis is specifically the diffusion of water across a selectively permeable membrane. So we're not really looking at other types of substances like ions or proteins or things like that. We just want to know which way water is going to move. So when we talk about osmosis, we have to discuss solutions. All right, solutions are mixtures where substances are completely dissolved within a fluid. The fluid that we're talking about, again, is water, okay? And the solute can be a number of different things. In our studies of cells, the solutes that we're looking at are going to be the things that are dissolved sort of within the cell. So all of those organelles and just different cellular material that would be inside the cytoplasm. And then other solutes that might be found in the environment of the cell. All right, so the solutes can be a lot of different kinds of things. But ultimately, what they're being dissolved in is always going to be water when we're talking about living systems. So there are three types of solutions, and they are called isotonic, hypotonic, and hypertonic solutions. And the reason why we call them tonics is because what's most important here is what we call the relative concentration. Okay, so we're going to keep coming back to this over and over and over again. If we talk about a cell, okay, here's a typical cell, and we're going to talk about the concentration found inside the cell versus the concentrations found out here in the environment around the cell. All right, so that's what we mean by relative concentration. How concentrated is the water inside the cell versus outside, and how concentrated are the solutes inside the cell versus outside. Okay, so ultimately it's this relative concentration that's going to determine the extent and the direction that the water is going to move. All right, so let's take a look into this. All right, we'll go over onto our next screen here. All right, if we break down a solution, okay, every solution is composed of two components. The solute, okay, which you can see here is the substance that's being dissolved. So in this case, we have copper sulfate that is going to act as the solute. And then it gets completely dissolved within what we call a solvent. Okay, the solvent is the fluid component of that mixture or solution. So it's the substance in which the solute gets dissolved. Now, in biology, our universal solvent is always going to be water. Okay, so universally, when we talk about solutions, we're talking about solutions that have a water solvent. Okay, so just keep that in mind as we continue to talk about what we mean by isotonic, hypotonic, and hypertonic. Okay, now this word iso means equal. So what that means is if we're going to compare the environment inside a cell versus the environment outside the cell, okay, in this image here you have your cell, okay, this is the cell's normal shape, and then out here in this white area is the environment around the cell. And if you look at the water molecules, which are pictured in these little blue dots, and the solute molecules, which are the slightly larger orangish yellow dots, their concentrations are pretty much the same both inside the cell and inside the solution. Okay, so that means that since we have equal concentrations, this is where the word equal comes into play, Since we have equal concentrations of our solutes and our water, then that means water is going to continue to move in all directions at the same rate. Remember, we talked about that with equilibrium. Okay, so water is going to enter the cell, as indicated by this arrow, and leave the cell at the exact same rate, which means the cell is not going to change shape. It's just going to stay its normal size. All right, now let's compare that to when we say we have a hypotonic solution. Okay, this prefix hypo means under. All right, and what is under is that the solution is undersaturated with solutes. Okay, so what this means is that the solute concentration inside this solution is very, very low. And since the solute concentration is low, then by comparison, that means that our water concentration, okay, remember I said I use these little brackets to indicate the word concentration, that means that our water concentration must be very, very high. If we don't have a lot of solute in the solution, then that means most of the solution is water. Okay, so the water concentration would be really, really high. So now let's take a closer look at our cell. Right, here's our cell. And now if you look inside this cell, you see far fewer water molecules and a lot more solute molecules. Versus the solution that cell is sitting in, you see a lot of water molecules and not very many solute molecules. So in this case, the environment is a hypotonic solution, right, where we have lots and lots of water outside the cell and not much water inside the cell. So water is going to move from where it's high outside the cell and it's going to go into the cell. And notice how these cells are a lot bigger than they were. Notice that these cells are a lot bigger than they were here in the isotonic. Here we kind of have, a slightly wrinkled cell membrane. Here we have, a perfectly square plant cell. All right, but if we compare that to these cells down here, this cell, the animal cell, has started to balloon up. Right, it's sort of gotten bigger, just like a balloon. And the same thing has been happening to this plant cell. It's sort of ballooning out, but it's not getting as big because of that rigid cell wall. All right, but look at how enormous this central vacuole has become. It's taking up most of the space inside that plant cell now. All right, so in hypotonic solutions, cells always tend to swell and get bigger in size because they're taking on water. Right? And then versus a hypertonic solution, this word hyper means over. So these are solutions that are overly saturated with solutes. So in this case, the solute concentration is really, really, really high, which means that our water concentration must be lower. Okay, so if most of the solution is solutes, that means that we don't have very much water in that solution. And look here. So here is our animal cell now. All right, you notice it's even more shrivelly than it was before. Right, and that's because we have more water inside the cell than outside. Right, and water always moves from where it's high to low. So in hypertonic solutions, cells will lose their water to the environment. And this causes cells to shrink in size. Right? You can see the same thing happening here with the plant cell. Now look, this little shriveled thing right there, that's where the central vacuole used to be. Right? But now it's completely empty because all of the water has left the cell, and the cell membrane has even pulled away from the cell wall. Look, this is the cell membrane right here. And it's pulled away from the cell wall, leaving behind this vacuumous space because the cell has shrunk so much. Right? Now something that I want to point out, since these are relative terms, hypertonic and hypotonic, notice here we said that the solution was hypotonic. If that's the case, then that must mean that what's inside the cell that environment is a hypertonic environment. Oops, hyper, I can spell well. There we go. Hypertonic solution. Right? And as a general rule, water will always move from a hypotonic to a hypertonic solution, because remember, hypotonic is where we have really high water. Hypertonic is where we have really low water. Right? So the same thing is true here. If the solution is acting as a hypertonic solution because it has all those solutes. There I go again. Why can I not spell hyper today? Oh my goodness. Hyper. Here we go. All right, if the solution is acting as a hypertonic solution, then the cell would be considered a hypotonic solution. And again, water goes from hypo to hyper, from where it's really high in water, hypotonic solution, to where it's really low in water, hypertonic solution. Okay, now there should be a link for you to see a video. Right, so you can click on that link. If you don't see the link there, I've also posted it to Canvas.